Okay, everybody, welcome to the InterSTEM Research Symposium for the today is April 2nd, 2023, and this is for the InterSTEM Research Program 2022-2023. Just some quick introductions on some of the teams that we've had this year. The three teams that have completed their research projects include the biology team with David, Carolyn, and Nishka, the biology mathematics team with Sneha and Lauren, and then finally the physics and mathematics team with Nicolette and Madison. All three of these teams have been working diligently over the past few months to complete an entire research paper on their own in their respective topics, and then also have created a presentation for you all today in today's symposium. And if you'd like to check out their research papers, they will be presented in a journal that will be published on the InterSTEM website in the coming weeks. Without further ado, let's get started with the biology team with David, Carolyn, and Nishka. And they are presenting on the effects of varying salt concentrations on DNA extraction from strawberries and bananas. I will stop share, and you all can go ahead and start sharing your screen now.
Can you all see the screen?
Yep, you're great.
DNA extraction is a process of vital importance to studying the genetic causes of disease. It's used in the development of diagnostics and drugs, and it's essential for carrying out forensic science, sequencing genomes, detecting bacteria and viruses in the environment, and determining paternity. For such a widely used process, it's important to understand how to optimize it. Various factors play a role in DNA extraction, one of them being salt concentration. My name is David, and over the course of the last 6 months, I, Nishka, and Caroline investigated the effect of DNA extraction on salt concentration. The process of DNA extraction occurs when something is used to lyse open a cell and separate DNA. Although prior research has revealed a variety of methods for DNA extraction, ranging from solid phase purification to capillary electrophoresis, for our purposes, we use the solution with a mixture of salt, water, and dish detergent. DNA is soluble in water, but insoluble in the presence of salt and alcohol. Salt helps to neutralize the charge of DNA's sugar phosphate backbone and makes it less hydrophilic, allowing it to dissociate from water more easily. Additionally, it stabilizes the DNA molecule after removing the proteins from it. By adding these to the water, the DNA precipitates into visible white pellets that can be collected. DNA extraction uses physical and chemical bonds from the sample that separates DNA from cell membranes, proteins, and other cellular components. Some of our additional background research also showed us that the fruit should be fresh and organically grown without the use of any pesticides or chemicals to ensure the purity of the DNA. We also purchase underripe fruits because they typically have the most intact DNA. Now that you've been provided with a basic background for DNA extraction, here's our hypothesis. We predict that a higher salt concentration will yield more DNA because it will neutralize more and separate from the organism. And strawberries will yield more DNA than bananas because they have more chromosomes in each cell.
Next, we'll be going over our methodology. To simplify the process of DNA extraction, it can be broken up into 4 main steps. First is homogenization, or creating the mixture needed. Next is lysis, which is when the cell wall is broken to allow the DNA to come out. Third is separation, where the DNA makes its way out of the homologized mixture. Lastly is precipitation, where the DNA accumulates at the top of a collection medium in a collectible form. To test our hypothesis, we first gently mashed our fruits in a Ziploc bag to prepare them for the extraction process without harming the DNA. Then approximately 100 grams of tap water was heated to 100 degrees Fahrenheit, since the greatest DNA yield is usually between 95 degrees Fahrenheit and 105 degrees Fahrenheit. Salt was then dissolved in the water. The amount of salt added was different for each trial to achieve different concentrations. To achieve a salt concentration of 0%, the control trial, we put no salt in the water. 0.5 teaspoons gave 2.08% concentration. 1 teaspoon was 4.17% concentration. 1.5 teaspoons was 6.25% concentration. And lastly, 2 teaspoons were 8.33% concentration. Then this saline solution was added to the Ziploc bag and mixed with the mashed fruit. This is to neutralize the charge of DNA, so it is less hydrophilic. The salt also removes the proteins from the DNA and keeps it dissolved in the water. About 80 milliliters of the mixture was then weighed out, and half a teaspoon of dish soap was mixed into it. Soap, being both hydrophilic and hydrophobic, has a compound that can remove fats and proteins, so it can break apart cells, which are held together by fats and proteins. Breaking this membrane releases the DNA in the cells. Now, the mixture is ready to be filtered into a graduated cylinder. The coffee filter was put inside a funnel over the graduated cylinder, and the mixture was poured through the filter. This was to block any larger chunks of fruit from entering the graduated cylinder and interfering with the extraction. After adding approximately 80 milliliters of the mixture to the cylinder and stirring it, 20 milliliters of the 91% isopropyl alcohol was added on top. This rubbing alcohol was added to make the DNA clump and form a visible white precipitate. As you can see in the picture, the alcohol is not soluble in the mixture, so it remains separate and provides a medium for the DNA to precipitate into. After 5 to 10 minutes, there was a white visible precipitate on top of the rubbing alcohol in the graduated cylinder. A popsicle stick was used to take this precipitate out and put it into a Ziploc bag for weighing. To get the highest accuracy, we used analytical balances to weigh the yield of extraction. Next, let's take a look at our results. As you can see in this graph, our results did not line up with our hypothesis. The bananas yielded significantly more DNA in all cases, and the control, which was expected to yield no DNA, somehow yielded the most. However, there is a general trend of yield increasing with salt concentration, so our hypothesis lines up in that regard.
Next, we'll be moving on to the discussion of our results. The results of the trials displayed a general trend of the DNA yield increasing with salt concentration. DNA extraction is made possible by precipitating the DNA out of a solution with water. Since water has a relatively weak positive charge, this is accomplished by providing a charged ion in the solution. Sodium is the perfect candidate for this. After the DNA dissociates from the water, it binds to the sodium, as they have opposing charges, and precipitates. The data begins to misalign with our hypothesis when analyzing the results of the control trials. In both strawberries and bananas, the control yielded a greater amount of DNA, despite having no sodium to allow for precipitation to occur. Although initially perplexed by these results, a repeat of the control yielded similar results. We are still unsure as to the exact cause of such a discrepancy, but it is likely due to the contents of the water solution. The control trial was run in New Delhi, India, while the experimental trials were done in the United States. As tap water was used by all parties, there is likely contamination in the water, especially in India. It is also possible that the differences in DNA yield was due to the variety of bananas used. The 91% isopropyl alcohol we used was a different color, suggesting a different chemical makeup. Although further testing is still required to narrow down a conclusion, we hypothesize that either the water or the isopropyl alcohol in India contained another chemical component with a positive charge that was able to neutralize the negatively charged DNA. It is also possible that the isopropyl alcohol alone allowed for such a high rate of extraction in the control. Although DNA is not soluble in the alcohol is miscible in water, meaning that it's able to form a homogeneous mixture. So as more is added, the solubility of DNA could potentially increase. The second half of our hypothesis was disproved, with bananas yielding significantly more DNA than the strawberries. The initial prediction that strawberries would allow for more DNA to precipitate stemmed from the fact that strawberries are octoploid, meaning that their cells have 8 duplicate copies of each chromosome. On the other hand, cultivated bananas are diploid, triploid, or tetraploid hybrids, meaning that their cells have 2, 3, or 4 sets of DNA. This makes the total chromosome count for strawberries 56 and the total chromosome count for bananas 33. This higher availability of DNA led us to believe that strawberries would yield significantly more than bananas, but the opposite occurred. This is likely due to our procedure. Since we stored the fruits in a cold environment, it led to the strawberries being overly firm, making it difficult to mash them in preparation for the extraction process. As such, The strawberries were likely not in a form that allowed for optimal extraction. Though the bananas were kept in a similar environment, they were still in the state that allowed for a greater amount of cells to be exposed through physical mashing.
In a replication of this experiment, both strawberries and bananas should be kept in refrigeration for an equal amount of time, and the uniformity of all consonants should be maintained. Distilled water should be used, and if researchers wish to test the effects of rubbing alcohol or dish detergent on DNA extraction, it should be done with separate procedures. In conclusion, this study aimed to explore the effects of salt concentration and the type of fruit on the DNA yield of bananas and strawberries. It was predicted that the DNA yield would increase with a higher salt concentration and that strawberries would yield the most DNA. And though this was somewhat accurate, the greatest DNA yield was at the salt concentration of 0, and bananas yielded significantly more DNA. Although our prediction had been supported by other trials, the results of 0% concentration were just vastly different from our predictions. We recommend that further research is conducted on the causes of the unexpected results of this study, such as the effect of the amount of mashing and regional discrepancies on DNA yield. Overall, conducting this project was an amazing experience for us all who share a passion for molecular biology, and we thank InterSTEM greatly for the opportunity. Thank you for listening.
Thank you all so much for presenting. If you can please stop screen sharing, and if any participants watching have any questions, feel free to put questions in the chat as I prepare to present the next team. All right, so the next team that will be presenting today in our symposium is the biology and mathematics team, and this is comprised of Sneha and Lauren, and they will be exploring alternatives to corticosteroids for the Duchenne muscular dystrophy, the promise of vamorolone, I believe is how I pronounce it. I'm sure you guys will be able to better pronounce everything, but without further ado, you guys can go ahead and share your screen.
Perfect. I'll go ahead and share that. Great. And can everyone see my screen?
Yep, you're good.
Okay, perfect. Good morning. My name is Sneha Jubi, and I was team leader for this project, which explored the alternatives to corticosteroids in Duchenne muscular dystrophy.
Hello, my name is Lauren, and I'm a co-author on this project. Duchenne muscular dystrophy, commonly referred to as DMD, is a rare genetic disorder that impacts approximately one baby per every 3,500 male births.
For patients impacted by DMD, there's a mutation in the gene that produces dystrophin, which is an essential protein in the dystrophin glycoprotein complex, also referred to as the DGC. The DGC is essential for muscular health because without the DGC, the structural link between the intracellular cytoskeleton and the extracellular matrix ultimately fails and results in improper or absent transmission of movement, as shown by the diagram on the right. The DGC is crucial to skeletal and cardiac muscle health and performance, so a diagnosis of DMD is devastating for patients.
Most impacted DMD patients commonly pass away before the age of 30, so treatment is primarily focused on slowing the rate of muscle degradation to retain mobility and quality of life. Unfortunately, while many treatments for DMD prevent the progression of the disease, they include side effects that substantially decrease quality of life. The primary treatment for DMD is a class of steroids called corticosteroids, a class of hormonal steroids created in the adrenal cortex. The medications prednisone and deflazacort are most standardly used, but side effects for these medications can include weight gain, loss of blood supply, mood swings, slowed growth, and the deterioration of bone or osteoporosis.
As a result, this literature review was conducted to evaluate side effects of treatments like corticosteroids, including prednisone and deflazacort, and compare those to the treatments using vamorolone, which is a synthetic steroid currently under production specifically created to target Duchenne muscular dystrophy.
For this literature review, multiple databases were utilized. Articles collected for this study were from English open source publications and credible scientific journals, with all articles being written in the past 10 years, with significant relevance to the progression of DMD or
We evaluated articles from databases including JAMA, PubMed, Google Scholar, ResearchGate, and more. Titles, abstracts, full papers, and references from these articles were used, and keyword searches were specifically utilized in order to refine the articles in these databases. As shown in these screenshots, the one on the left is from JAMA and the one on the right is from PubMed. We use specific keyword searches, such as deflazacort medication for dystrophy, prednisone impact on muscle degeneration, and vamorolone in Duchenne muscular dystrophy to refine our results.
So in these research papers, various clinical studies conducted their trials in a multitude of ways. However, not many papers directly compared the corticosteroids deflazacort and vamorolone, but there was a considerable amount of research done comparing prednisone and vamorolone. Two methods that we specifically looked for in previous studies and research were analysis for creatine kinase KM, a protein that indicates sarcolemma damage, or through patient ambulation.
Overall, multiple credible articles were referenced throughout this process to measure the impact of different medications on the progress of Duchenne muscular dystrophy and the negative side effects as well.
So in terms of patient ambulation, in one specific study, the authors evaluated the efficacy of treatment and the progression of DMD through measurements known as TT stand, TTRW, and TT climb, which measured the time a patient needed to stand, the time a patient needed to run or walk 10 meters, and the time a patient needed to climb as labeled on our graph below.
Our results, generally speaking, found that patients treated with vamorolone had much fewer side effects than those treated with different dosages of both prednisone and deflazacort, which are two corticosteroids. This specific study analyzed 4 key side effects, which was a Cushingoid appearance, as demonstrated by the diagram on the left, this and hirsutism, as well as behavioral changes while on one of these 3 drugs.
Overall, we found that while patients being prescribed vamorolone certainly experienced side effects, these effects were substantially less severe than the side effects of patients on different medications, while still maintaining a similar prognosis.
Overall, compiling this literature review did answer our initial research question, which was, in Duchenne muscular dystrophy, what is the effectiveness of corticosteroids like deflazacort and prednisone compared to alternative treatments such as vamorolone? Our result ultimately demonstrated that corticosteroids were an effective treatment for DMD and that vamorolone can achieve similar results with less severe side effects.
While this research project typically analyzed very recent research articles and publications, there was not too much research that directly compared the 3 corticosteroids, which could have limited the data.
Another limitation was that the clinical studies that we evaluated throughout this literature review process did not always have the most diversity in clinical participants, with one specific study having patient data from populations that were Asian, white, and Hispanic, but lacking any data at all from patient populations that were Native American or Black. In the future, we hope that more research will be conducted in these underrepresented areas of medicine in order to ensure equity in clinical trials. Generally, our findings did align with the findings of other researchers, showing that vamorolone typically had similar rates of slowing down the progression of Duchenne muscular dystrophy as other corticosteroids, but did so with less severe side effects.
Ultimately, these findings have considerable implications for society because patients who have been diagnosed with DMD may look into specific treatments and find that certain alternatives such as vamorolone may be safer and preferred by not worsening their quality of life through harsh side effects. To conclude, Duchenne muscular dystrophy is a rare genetic X-linked disorder that causes progressive muscle degeneration. While there are numerous corticosteroid treatments for DMD that slow rates of disease progression, standard steroid treatments such as prednisone and deflazacort may cause harsher side effects than vamorolone.
In the future, research may be conducted analyzing how vamorolone compares to other treatments, such as allopathic medicines and occupational therapies. Or a study could be done analyzing the accessibility of these three drugs to patients of different socioeconomic groups, and how this is an early indicator for overall health and well-being. Overall, Lauren and I really enjoyed working with InterSTEM on this research paper. That concludes our presentation and our paper. Thank you all so much.
Thank you so much for that presentation. And thank you for stopping your screen share. Again, if anybody has any questions, please feel free to put those in the chat. And then I will share my screen again. Okay. Thank you again. And the next team that unfortunately is not able to present again, but they did complete a research paper, is the physics and math team by Nicolette and Madison, and they were investigating the features of hypersonic cruise missiles and hypersonic glide vehicles that renders them undetectable on radars. Their research paper will be featured in the InterSTEM Research Journal that will be published in the coming weeks. So I do recommend that you all check out their research paper as well. As for that concludes our final session for the InterSTEM Research Program. A huge congratulations to all the teams that completed their projects and their presentations. You did a phenomenal job.